Let's see. Key is not here. Boggs is here and WBSU here. Bonner is not here. Clay is not here. Dawson is not here. Ellis is not here. Paula is not here. Green is not here. Harrison is here. Caleb's here. WBSU here. James is not here. Kesterson's not here. King is not here. Lewis is not here. Lindick is here. Is that WBSU? Yep. NWBSU here. Morrison is not here. Grace is not here. Ward is not here. Wilcox is not here. Wilder is not here. Parks is not here. So I got about 5 minutes. Let's see who's online. James is online. Don't forget to send me your attendance words, everybody, online. Otherwise, you're considered absent. Okay. Lewis is online. Ward is online. Again, don't forget to send me the words. Just because you're logged in doesn't mean you're awake and paying attention. Grace is online. Green is online. Wilder is online. Morrison is online. Ellis is here, right? Yep. Ellis is physically here. Dawson back there?
Yes, sir.
All right. So Dawson is here. Does anybody have any announcements on your end while we're waiting? Anything for about athletics or student activities or just place you work? Anything you want to say? Okay. Let's see. Parks is online. The online people can hear me. So I'll give you some reminders then, since we've got a little bit more than a minute left. So again, Veterans Day, which is Monday, November 11th. We're only having an online meeting. And you can come here if you want, but I won't be here. We're just doing it online only. I guess just go ahead and plan for the worst. If something happens and you log on and I'm not there, just don't worry about it. I will come up. I've got hundreds of videos. So if something happens to me or my computer or something, I'll just send you out a video that you can watch instead of meeting on Monday. Now that we've had a plan B, nothing's going to go wrong. But if I did have a plan B, something would have gone wrong. So there we go. We've now made sure we're safe there. Wilcox is here. So yeah, don't forget that again. Online only on Monday, November 11th. And also the Friday before that, apparently, it's a reminder. My kids have a Veterans Day thing going on at their school, so I have to go to that. So I'll not be having office hours Friday before Veterans Day. Of course, if you need me, as usual, send me an email or call my office or something. Say, hey, can you meet me online? And once I'm done with that thing, I'll make some time to meet with you. Which is another good time to remind you, if you need my help, get it, right? I'm online a lot. Just come meet me online. You can share your screen. I can share my screen. We can talk about assignments, and I can help you with them. Speaking of which, today's lab is completely virtual, so I don't care if you show up or not. You can come in and do it here with my help if you need it. You can meet me online. You get my help if you need it, or you can just completely do it on your own. However you want to do it. Just make sure you have it done by Friday. It's pretty simple, very simple lab. That's it for announcements and reminders. It's 8 o'clock, so let's get started. We should be already recording. Hopefully the people online can hear me. The first word, the official word for attendance. Let's see. It's going to be RNA. The word I just boxed in. That's the first official word for attendance. So what we've talked about so far is we've had like a big overview of how we go from DNA to RNA to protein. And by now you should know that going from DNA to RNA is called translation. Going from RNA to protein is called, DNA to RNA is called transcription. And going from RNA to protein is called translation. You should know that by now. And we had a big overview, talked like the big picture of how that works. Gave you some specific information about how nucleotides work and how they dictate the amino acids. And we talked about the universality of the genetic code. Now we have that background information. We're going to go back and look at it in more detail. And again, to remind you, I'm doing this a little bit out of order than your textbook. This is the way the old textbook did it. And I like their way better. I like the big picture and then diving back in for a deeper look at it. And I'll remind you, for independent work, anything I say or anything in the textbook that is a perfect independent work topic, go to your online textbook, copy something, paste it in a Google document and say, the textbook says this about transcription, right? And then go to a different source and say, but this source says, copy it, paste it, says this about transcription. And then just write a few sentences about compare and contrast. Like maybe they're exactly the same. You say, well, they pretty much say the same thing in the same way. Or if they're a little bit different, just compare and contrast, right? So you want to directly quote one source, directly quote the other, and then you write your own words how those two things are the same or different. So keep that in mind for everything we're doing as we go through these lectures, because you guys really need your independent work. All right, went ahead and put your grades in. So you see your grades probably dropped, at least on WBSU Online, because I put your grades in. One person went up because they've been done with independent work for weeks now. So, and also remember, anything you get over 100 points is then considered extra credit. Anyway, here we go. Transcription, going from DNA to RNA. And again, like I said, when you're studying this, first remember that first. Remember that it's called, going from DNA to RNA is called transcription. Then you can start adding these details that I'm about to give you. As you already know, the DNA strands, actually, no, you don't know that, I'm sorry. This is new, yeah, I have told you that, sorry. So the DNA strands separate when this happens, just like they do for DNA replication. Remember we talked about how DNA replication works, where the DNA polymerase attaches to the strand of DNA, and the strand is opened up because those hydrogen bonds are really weak, and then the DNA polymerase can read the code and bring in the complementary DNA nucleotides, right? And in a sense, this is very similar, and you'll see how, right? So once again, the DNA strand has to open up so that code on the inside can be read. So far, this is the same as DNA replication. Once that DNA strand is opened up, and this is going to be a horrible drawing of it, but try to draw it. That, okay. Yeah, that looks like garbage, sorry. But anyway, once it's opened up like that, one of these sides is going to serve as the template to make the RNA, and the other one's just not used, right? So one's used, one's not used. Not that I'm going to ask you that on the exam, but that's how that works. Sort of like it happens when we do DNA replication, RNA nucleotides take their place along the DNA template by forming hydrogen bonds with the DNA nucleotide bases, which is, again, basically the same thing I said for our DNA replication. We open it up, the cellular machinery reads the code and says, all right, I need this next nucleotide, this next nucleotide. So far as we're concerned, this is exactly the same as DNA replication, except instead of bringing in DNA nucleotides, we are now bringing in RNA nucleotides, right? Because we're not trying to duplicate the DNA strand. We're trying to transcribe the DNA. We're trying to make a strand of RNA that is complementary to the strand of DNA. So again, so far, everything is exactly the same as I told you, except we're not dealing with DNA nucleotides, we're bringing in RNA nucleotides. Are there any questions so far? All right. Once again, as you already know, RNA nucleotides follow the same base pairing rules. So A's and T's go together, C's and G's go together, except keep in mind for RNA, there is no T, there's a U instead. So again, if this is a strand of DNA over here, and this is a strand of RNA, if DNA has a C, RNA would have a G. DNA has a G, RNA would have a C. If DNA were to have an A, it should be a T, right? T's go with A. But again, RNA doesn't have a T, so it has a U instead. You would substitute the U for the T. And of course, if DNA had a T, then RNA would have an A. Are there any questions about that? It's not the first time you've seen that. It's important. Because remember, I will give you a strand of DNA on the exam and say, I need you to transcribe this. And again, for this multiple choice exam, when you're looking at your multiple choices, if any of them have a T on the transcription question, then you know that's not the right answer. Because RNA, when I'm asking you to transcribe something, I'm asking you to make a strand of RNA based on the code from the DNA. So if you're making a strand of RNA, then any answer that has a T in it is incorrect because it is not RNA. So here's some more new information. Like I said, this is very similar, as far as we're concerned, very similar to DNA replication, except, once again, we're not bringing in DNA nucleotides, we're bringing in RNA nucleotides. And also, here's another one for you. We are no longer using something called DNA polymerase, we are using RNA polymerase. Someone's going to miss those questions on the exam, and it's going to make me sad, because this is probably one of the easiest questions on the exam. There's only two enzymes we've talked about in this section of the semester. DNA polymerase and RNA polymerase. DNA polymerase makes DNA. RNA polymerase makes RNA. It's just that simple. It's not a trick question, so when you get that on the exam, please don't miss it. And this is kind of what it looks like, right? You can see sort of, this is a strand of DNA at the bottom. You can see the RNA polymerase is attached to it. You can see the DNA is kind of opened up a little bit, and then this is the new strand of RNA that the RNA polymerase is making. You might also notice that there's this RNA polymerase is different colors. And if this was I don't know how to put it. If this class was for biology majors, or if it was not 100 level, then you would need to know that technically RNA polymerase is it's kind of like a team of proteins. But as far as we're concerned, you can just consider it one protein. Just so you know in the back of your mind, it's not actually just one simple protein, it's a team of proteins that do this. Here's a picture of what it looks like. This is from the old textbook. But again, this should look very similar from what we talked about when we talked about DNA replication. Once again, we are opening up that DNA strand to read the code. Once again, there's something this is bringing in nucleotides. Like there's a T, all right, better bring in an A. There's an A, better bring in a U. There's another A, better bring in another U, right? So that's how it works. This RNA polymerase reads the code and brings in the complementary RNA nucleotides and hydrogen bonds them, covalently bonds them to that growing strand of RNA. Pretty simple, right? Any questions about that picture? Okay. Here's where it gets a little bit more complicated. Transcription itself is broken down into 3 stages, and you need to know these 3 stages. Now, the names of them, know what order they are in. They are initiation of transcription, RNA elongation, and termination of transcription. Notice I put some of those in bold. So if you were to make a flashcard, you could put something like, what are the 3 stages of RNA transcription? And you could just shorten that to initiation, elongation, termination. Because I'll go ahead and tell you now on the exam, I'm not going to try to trick you. So you can just memorize initiation, elongation, termination. And here's a little spoiler for you. When we talk about translation, it is also the same steps if you put it in bold. It'll be initiation of translation, and it'll be elongation of the polypeptide, and then termination of translation. But again, if you can just remember initiation, elongation, termination, that's perfect. And again, in my opinion, the best way to study this is memorize this big picture, then start adding the details. So once you have it memorized, what, initiation, elongation, termination, then start memorizing what each one is, because we will start them or talk about them separately. All right, does anybody remember, because you will need to know this for the exam, I don't expect you to remember yet because I've only told you once, and I would assume you haven't been doing a bunch of studying since Monday. But does anybody remember where on the DNA strand DNA replication starts? There was a word for it. Remember, there was a bunch of those spots on the DNA strand where DNA replication takes place or starts. Do you remember what that was called? All right, that's okay. You'll need to know before the exam, but just to remind you, it's called origins of replication. Here, similar thing, it's just a different name. So the start transcribing signal is a DNA sequence called the promoter. So before any gene is this area before it that has the A's, the T's, the C's, and the G's, and that's called a promoter. And if you really got into it, it's actually quite complicated, but for this class, it's not complicated. All you need to know is that's where this whole process starts. Where does transcription start? It starts at the promoter. Even more specifically, where does initiation of transcription start? It starts at the promoter. Again, like I already said, it's located at the beginning of the gene, which makes sense because if something's going to attach to the DNA strand and read the gene, obviously it's going to need to be before the gene, right? Or at the beginning of the gene. And obviously, again, logically, that is going to be where the RNA polymerase attaches. This should be all, I won't say common sense, but logical. Of course, these words might be new to you, but the idea of what's happening should make sense to you, even if this is the first time you've seen it. So then, that being said, what exactly is initiation? It is when the RNA polymerase attaches to the promoter. That's pretty much it, right? So that is what initiation of transcription is. The RNA polymerase attaches to the promoter, and that's what we call initiation. So knowing what the goal of initiation is, of transcription is, if this is step one, and also I already gave you the name of the next step, so let's see if you can figure out, what do you think is going to happen next? If step one is the RNA polymerase just attaching to the DNA, what do you think is going to happen next? Logically, this should make sense to you. This isn't that hard. I think somebody will be able to figure it out for a little bit of extra credit. What must happen? What logically must be the next step? The RNA polymerase goes backwards and disintegrates. No, I'm just kidding. That's not what happens, right? That would not be logical. Think about it. This is one thing that could happen. Don't overthink it. Step one, the thing attaches right before the gene. What's going to happen next? Logically, what has to happen next? Doesn't have to, but it shouldn't be that hard as long as you don't overthink it. Do you think the RNA polymerase, turns into ATP and then does photosynthesis? No, right? Think what must happen. Imagine it. It's attaching here on the DNA strand. The purpose of this thing is to make a strand of RNA by reading the code in the DNA. We already did that. So what must happen next? Oh, you got it? Okay. We're a little bit ahead, so I'm not trying to waste time, but I really want you guys to wake up. Just think about it. Don't be shy. What must happen next?
What's the question?
All right, I'll back up a little bit. This is good. So we're talking about transcription, which is where we go from DNA to RNA. And I've told them that transcription is broken down into 3 stages. Initiation of transcription, which is this right here, where the RNA polymerase attaches to the DNA strand. Then there's elongation, and then there's termination. And I was telling him, what's happened here is the machine that does the reading of the DNA that makes the RNA strand, which is what we're after, in this first stage, it just attaches to the DNA strand. So what logically must happen next? Just give it a few more seconds to see if anyone gets it. Hopefully you guys know you're just being shy and don't want to sound dumb. And it's okay, none of you guys are biology majors, so don't feel bad if you get it wrong.
Will they have to bond together?
They bonded, yep, that's part of this. Yep, it bonds in this stage, step one, it's bonded to the DNA strand. And the next stage is called elongation.
That's where it makes the helix.
This, yeah, basically. Because remember now though, that I want to call it a helix, but in this case, it makes the RNA strand. So logically, after the thing attaches, it moves forward and starts reading the code, and as the name suggests, it's elongating this strand of RNA. So it's making the strand of RNA. So this is like the meat and potatoes of the 3 stages. This is where the good stuff happens, right? This is what we're after. So this is when the RNA polymerase moves down the DNA, like we've already seen in that previous slide. It's reading the code, it's opening the DNA up. It's reading the code. It's bringing in RNA nucleotides one after the other, covalently bonding them to the growing strand. That's it. And then also, now some of this wouldn't necessarily be logical. Like I wouldn't expect you to be able to guess this, but after the DNA strand has been read, the DNA then comes back together. I probably won't even ask you that. And you wouldn't even, I guess I wouldn't even necessarily expect you to logically guess that the RNA kind of peels away from the DNA, but now you know. But yeah, the rest of it, that should have been, I think, pretty logical, right? If step 1 is the thing attaches to the DNA, step 2 must be it keeps going and it's reading the code and it's building that DNA strand. All right, now given the name of the 3rd stage, just using basic logic, and I'm not going to wait that long for this one, but can anyone guess what happens in the 3rd and final stage? Okay, so termination means we're terminating this whole process. So basically, it's kind of like the opposite of initiation. It reads a certain signal, which is called the terminator, right? So instead of the promoter, it's called the terminator. When it reads the terminator, the RNA polymerase is like, oh, job is done here. So all the parts just separate from each other. DNA is back to, it's closed back together. The RNA polymerase is gone. The RNA itself is now ready to move on to go to translation. So are there any questions about any of the 3 stages before I show you the next slide, which is a picture of all 3 stages together? Okay, nice and easy, right? So this is all 3 together. So again, this is transcription. The purpose of transcription is to make a strand of RNA that's basically a copy of the DNA, right? So the RNA polymerase attaches in a place called the promoter. That's what initiation is. Then it moves forward, opening up the DNA strand, reading the code, bringing in nucleotide after nucleotide to make this strand of RNA longer. That's called elongation. Finally, that RNA polymerase reaches what's called the terminator, which is a strand, a code of DNA that says, all right, the job's done. This is where the gene ends. So all the parts go their own way, right? The DNA is closed back up. RNA polymerase is ready to go do it again for another strand of DNA. And this RNA is now ready to move on to the next stage. Any questions about this slide? All right, let's see. The next word for attendance, I'm going to say corn. Because to me, that RNA polymerase looks like somebody tried to draw a kernel of corn and did a bad job. So corn with a C is the next word for attendance. Any questions? Okay, again, if you download a PowerPoint, you can watch this little animation that shows transcription in action. Now, if you remember, like I said, after transcription comes translation. However, we have to make it just a little bit harder for students. So that first, there has to be this in-between stage, right? So the RNA that we just got finished talking about, it's not actually ready to go into translation. But we need to talk about that kind of in-between zone. Sort of like when we talked about cellular respiration. Remember we talked about glycolysis takes glucose and breaks it down into 2 molecules of pyruvate, and then that goes into the citric acid cycle. And I was like, but the pyruvate can't go into the citric acid cycle. Technically, there's this in-between spot where we change the pyruvate to acetyl-CoA, right? Same thing here. I'm saying we are going from transcription to translation, but first there's this in-between spot. First, we have to process this RNA. But I also will point this out. In prokaryotes, everything I'm about to teach you does not happen. So for the more basic things like bacteria, all this doesn't happen, right? This is only in eukaryotes. And remind you, eukaryotes are the more complicated organisms, like humans, and other animals, plants, fungi, protists, the more complicated organisms, right? So That being said, oh wait, actually, here's a new word for you. I keep saying DNA to RNA, DNA to RNA, DNA to RNA, 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 right? Now I'm telling you there's actually different types of RNA, and you're going to need to know the difference between them on the exam. So technically, all this RNA I've been teaching you about this whole time, it's called mRNA. It is the messenger RNA. Hopefully that's easy to remember. Because when we're talking about transcription, again, remember, think of it as a non-biology term. The word transcribe means to write something down. So it makes sense that if you're writing down, if you're transcribing the message that's in the DNA into RNA, then that would be the messenger RNA, right? This is the RNA that is bringing the message. That is the molecule that will be translated into protein. The message that will be translated into protein. So here's some more stuff you might need to know for the exam. In eukaryotes, transcription happens in the nucleus. I've already told you that, so that's not new information, just a reminder. And of course, in prokaryotes, they don't even have a nucleus, so it just happens right away. In eukaryotes, the RNA is modified before leaving the nucleus and going into the cytoplasm. And that's what we're about to talk about in the next few slides. We're going to talk about that modification. Any questions so far? Again, in my opinion, and everybody's brain is different, but for me, the way I remember this as a student was thinking about the language aspect of it, right? From going from DNA to RNA, we're in the same language. It's the nucleic acid language. So we're literally just writing down a message, right? Writing down a copy. When I think writing, I think of transcription. And then when we go from RNA, specifically mRNA, to protein, to polypeptides, that's when we're translating, right? We're going from one language to the other. We're going from the amino acid language to the language of, we're going from the nucleic acid language to the language of polypeptides. But anyway, let's talk about that modification. The first one you need to know about is something called cap and tail. Cap and tail is one of the types of processing we're going to talk about. And of course, if you were to look at a different textbook, they might first tell you about cap and tell you what the cap is, and then specifically tell you about tail. But in this case, we're just going to lump them together for this 100 level non-biology major course. Cap and tail, one type of RNA processing. It's basically the addition of extra nucleotides to the ends of both tips of the RNA. This protects it from attack by enzymes and helps ribosomes, which I'm going to teach you about later, recognize the RNA as mRNA. So let me unpack those statements really quickly. So to remind you, well, not remind you, I haven't taught you yet, but later in this chapter, you're going to learn how viruses work. Basically, a virus is something that can't reproduce itself. So it puts its DNA or its RNA, depending on what kind of virus it is, into your cell. And your cells, hopefully, can recognize it as foreign, and then they attack it and destroy it. Of course, that doesn't always work, otherwise there wouldn't be any viruses, right? So that's what we mean by protected from attack by enzymes, right? We don't want random nucleotides. We don't want random DNA or random RNA just hanging out in our cells. It's not safe. So if we are about to put some RNA into our cells, right, out of our nucleus into our cells, then we want to protect it. So that's what we mean by protected from attack by enzymes. A good independent work question, and again, don't do it now, pay attention to the lecture, but for 1 point, you could email me, it wouldn't even need to be a paper because it's 1 point, right? What enzymes would attack the RNA, right? So what are the enzymes that are doing the attacking? What enzymes are being protected against? For one question, just look it up, copy it, paste it. So according to this source, here's the answer to your question, right? It doesn't even require you to write anything other than according to this source, here's the answer to your question. That's 1 point of independent work. And again, if you wanted to, you could write about it as a paper where you get more than 1 point, depending on the quality and length of the paper. But anyway, so that's the one thing. Let's also unpack this other part. It helps the ribosomes recognize the RNA as mRNA. So to remind you, like I said in the last slide, there are different types of RNA, right? And this just happens to be the first one I taught you. It's called mRNA. So every RNA is different. They all have different jobs. So we need this RNA to be recognized as mRNA. That's what that part means. The other part, I'm about to teach you this in more detail, but it says ribosomes recognize it. Again, I'm going to formally introduce you to this term later, but right now I'll just tell you ribosomes are the thing that do the translating. Those are the things that read the code on the RNA and build the protein. That's what that means, right? So this cap and tail, once again, protects it and also helps the cellular machinery recognize it as the mRNA. Any questions about that side? Notice too, and I don't remember if your textbook does this or not, but technically the cap is at the 5' end. Technically the tail is at the 3' end. And technically it's a poly A tail, meaning that's just a bunch of A's. But you do not need to know that. Just know that cap and tail is one of the types of RNA processing that protects it and helps it be recognized as mRNA. I don't care how deep your textbook gets into it. I'm telling you, that's all you need to know for the exam. So if there's no questions, we move forward to the other type of processing, and it is called RNA splicing. Here's where I'm going to teach you some more words. I used to hate these words as a freshman because they always confuse me. They sound similar, and I can see either one describing the other one. But let's talk about it really quickly. RNA splicing, as you might imagine, it's just splicing of the RNA, meaning we cut some parts off, like the parts in between, and then splice them together. So I don't know if any of you have ever worked with electronics, but that's kind of like if you're splicing a wire, that's what we mean, right? You're cutting a bad part of the wire out, you kind of put the other two good parts together, and you splice it. Same thing here. But the more formal way of saying that in a biology class is this. RNA splicing removes non-coding nucleotides from those that do code for amino acids. And as far as we're concerned in this class, and this is not a true statement, but as far as the exam is concerned and you studying is concerned, basically I could rewrite that bullet point to say it removes the junk DNA. Now granted, it's not actually junk DNA, it serves a purpose, but as far as we're concerned in this 100 level non-biology major class, it's just junk. Here's where it gets tricky though, and you do need to know the difference between these two for the exam. The junk DNA, the non-coding stuff, it's called introns. And the stuff that is used, so yeah, once again, the non-coding DNA, the stuff that doesn't code for anything, the stuff that I'm calling junk for the purposes of this course, that's called introns. The stuff that we actually use and that stays, that does not get cut out during RNA splicing, that's the stuff that is coding, and that is called exons. So you need to know the difference between the exons and introns. And that was always hard for me, and I'm not going to say why, because if your brain didn't think of it, I don't want to put that in your brain because that might make it hard for you. But here's how the proper way to remember it. Exons, that's the DNA that's expressed. That's the code in the DNA that gets expressed as a protein. The introns, again, just to help you remember it for the exam, is just the junk in between the stuff that's expressed. So exons are expressed. The introns are the junk in between that gets cut out. And there's going to be another slide. We're going to talk about this a little bit more. But are there any questions so far about RNA splicing? All right. Notice too that I keep saying mRNA. Remember, that's the message, right? That's the mRNA is the stuff that we just made in the process of transcription. And mRNA is the stuff that's going to be used in translation to make the proteins. But technically, before it goes through RNA splicing and before it goes through the cap and tail, it's called pre-mRNA. I put a big old X through that. I don't remember how deep your textbook goes, but I just want you to know I'm not going to distinguish between pre-mRNA and mRNA. As far as you are concerned for this exam, mRNA is the stuff that's made from transcription and the stuff that's used in translation to make proteins. I'm not going to distinguish between pre-mRNA and mRNA. Anyway, there's no questions about this.
Is the intron recycled in our bodies, or is it took out as waste?
You know what? I don't even know that. Hopefully I was never taught that and I didn't forget. I hope it's that I was never taught as opposed to I forgot. That's a good question. Look that up for independent work. And that is something I'm going to look up now. Because what happens? Here's what I'm thinking. This is a hypothesis. Again, I'm saying that's the junk DNA. Even though it does serve a purpose, in this specific instance, if it's getting cut out, in this case, it is junk DNA. So the question is, what happens with it, right? So are there enzymes that break down that slice of mRNA. So then you have random floating nucleotides. So then those nucleotides can be used again. It's a great question. Yeah, and I think I used to know the answer. Because there's a bunch of different types of RNAs that we haven't talked about, and that we won't talk about this in this class. I'll go ahead and tell you, as a spoiler, there's mRNA, which is what we're talking about now, tRNA, which I'm going to teach you about later, and rRNA, which I'm going to teach you about later. That's the three that you're going to learn. But there's a bunch of other different types of RNA that have very specific jobs, and I'm trying to remember if that's part of where they came from. But great question. Matter of fact, when you send me your words today, remind me that you asked me that question for a little extra credit. Any other questions before we talk a little bit more about RNA splicing? All right. So the exons and the the exons and the introns, all of it, are transcribed when we go from DNA to RNA, right? So when we talked about transcription, it's all transcribed. That's why we have to do the splicing, right? Because the whole thing is transcribed. It's then only when it's spliced do we get rid of the introns. Anyway, any questions? And still, we're going to get a little bit deeper into RNA splicing. Are there any questions so far? All right, here's some interesting numbers for you that you don't need to know for the exam. But RNA splicing plays a significant role in humans because it allows our about 21,000 genes to produce many thousands more polypeptides. Think about that. I keep saying that genes are instructions on how to build polypeptides for proteins, right? Let's see, you can put / proteins, or PR for proteins, right? So we have about 21,000 genes, which is another good independent work question for 1 point. Just email me the answer. Exactly how many genes do we think we have now? Of course, this was written about 10 years ago. So maybe you get an exact answer. Or maybe you'll find an answer that's slightly different. So exactly how many genes do we have now? Anyway, the point is, we have about 21,000 genes, but we have many more proteins. So how does that work? If the proteins, every single protein in our body, other than that's something you may have eaten, every single protein that's built in our cells was built because we have instructions on how to build them. So how is it that we only have 21,000 sets of instructions, but we have many thousands more proteins? And that's also another independent work topic, if you want, or question for 1 point. Many thousands more? How many? How many polypeptides do we make? But anyway, again, the big picture here is how in the world do we only have 21,000 sets of instructions of how to build things, but we have thousands more things that are built? I'll term it that way. And I'll actually come back to that because unfortunately when I made this PowerPoint, I put this slide out of order. So this slide should have been before this one. But anyway, here's what RNA transcription looks like. Here's what RNA splicing looks like. So again, there's the DNA that has all of it. Exons, introns, it has all of it. During transcription, the RNA polymerase reads that code and copies it directly, right? It's a perfect copy, if you want to call it that. It goes from DNA to RNA. But it copies everything, the exons and the introns. Then we add the cap and the tail. Then we cut out the introns because we don't need them. We splice that DNA back together. And now we have finally the mRNA that's ready to leave the nucleus and be translated into a protein. But anyway, like I said, that slide should have came before this one that we just talked about. And again, I'll remind you, how in the world did we get from 21,000 genes to many thousands more proteins? How does that work? And that is something called alternative splicing. So what this means is, and now it's going to get even more complicated. So here we have like this hypothetical gene has 1, 2, 3, 4, 5 exons. So you can see they're kind of spaced apart slightly differently. You can see some exons are bigger than other exons. And then here we have the in-between stuff, which is not even labeled in this picture. That's the introns. So what I just taught you was regular RNA splicing, right? So we get rid of all the introns, and that's what we have here, right? This is the kind of RNA splicing that I taught you. We just got rid of all the introns. So there we go. Exon 1 is spliced to exon 2, which is spliced to exon 3, which is spliced to exon 4, which is spliced to exon 5, right? We got rid of all the introns. But then we have something called alternative splicing, which is why I kept saying, emphasizing the whole junk DNA. It's not technically junk. Sometimes it's used. Actually, this picture doesn't even get that deep into that, so forget I even said that. So in this version, so instead of, again, we have exon 1, 2, 3, 4, 5, the whole thing all put together. In this version, we have exon 1, 2, 4, and 5. So not only did we splice out all the introns here, but we also left out exon 3. And in this version, let's see, we have 1, 2, 3, 5. So in this version, not only did we cut out all the introns, but we also cut out exon 4. And not that I expect you to know what these pictures mean down here, but again, form and function, you can see that these things have very different shapes, right? These proteins, remember, proteins are all about the shape. Even though those symbols probably don't make much sense to you, you can see that these three things look very different to each other, right? From each other, they're different. All because we have alternative splicing. And now we have 3 different proteins based on this one gene. And of course, keep in mind too, this is oversimplified. I think most genes have more than 5 exons, right? So imagine all the different possibilities, which is another good independent work question that you can look up if you want. Typical gene has how many exons? Again, copy it, paste it, email it. Doesn't even need, don't even put it, if you put it in a document that I assume you're writing a paper, it'll be created based on how long it is. But if you just want the one easy point, email it to me. But yes, how many exons is a typical gene? For that matter, how many nucleotides is a typical gene? Anyway, any questions about this slide? Any questions about RNA splicing or alternative splicing? Okay, then, and that question, is there any questions at all about transcription or this in-between zone between transcription and translation? Because if there's not, we're going to jump into translation. Okay, then let's jump into translation. And again, teaching out of the old textbook for this chapter. I like the way they did it. So before we talk about the process of translation, that textbook talks about the parts. It introduces you to the parts and what they do. And then once you're familiar with the parts, then they're like, all right, this is how they all work together. So let's do that. Let's talk about the parts. And the next word for attendance, since it's up there, actually, you know what? No. The next word for attendance is corn with a K, like the band Korn. So not with a C, corn with a K. Anyway, let's talk about translation. The players, the parts. The first main ingredient is the stuff we've already talked about, which is the mRNA, right? That's one of the most important part. Well, I guess none of it would work without any, they're all important. You just need them all. But might as well, this should be the first one we talked about because we've already talked about, right? So the mRNA is the message, right? It's the thing that says this is what needs to be translated into protein. So speaking of which, then there's also the machine. I need to change this PowerPoint. Anyway, there's the machine that actually does the translation, which I've introduced you to before. And that machine is called ribosome. So the ribosome is the thing that actually reads the code on the RNA and translates that and builds the protein. I'm probably not going to ask you this, but yes, it does require energy. Requires energy usually in the form of ATP. There are other sources of energy, but for this 100 level non-biology major class, we don't talk about it. But this is something you could look up, and I think it reminds me of a question maybe Kesterson asked in a previous lecture, like how much energy? So I'm saying, just to simplify it, we need ATP. Like how much ATP do we need to build the typical protein? Or if you find, depending on the source you use, maybe it's really specific. Maybe it says for each nucleotide, it takes this many ATP. I don't know, you can look it up if you want. Obviously, it's not important, otherwise I would tell you. Anyway, again, so we have the mRNA, we have the machine that does the translation, which is called ribosomes. We need energy. And then we also need another type of RNA called the transfer RNA. So now I've taught you 2 of the 3 types of RNA that you need to know about. mRNA, which is the one we've been talking about, and now this new one called tRNA, transfer RNA. Now, this is only slightly logical, so I'm not going to sit and wait like I did with the other stuff, because I'd be really surprised if anyone guesses this, but it's not impossible to guess. What do you think, given what we're trying to accomplish here in translation, and given that I didn't put this on the board yet, what do you think that tRNA is transferring into this process? What is it transferring? And I'll go ahead and make it a little bit easier to because we have some other stuff. So you might think, oh, it's transferring the machine in. So no, transfer RNA doesn't transfer in the ribosomes. It doesn't transfer in the enzymes. It doesn't transfer in the energy. It doesn't transfer in the mRNA. There's something on this board, or there's something not on this slide that we need to build polypeptides. And that's what it's transferring in. So this is almost like a throwback, because you need to remember back in chapter 2, when we talked about what polypeptides are made of, what proteins are made of. So what do you think is that's bringing in? The only thing that's not on this slide that we need to build protein.
Code?
We have the code, great guess. The code is written in the mRNA. So what do we need? And actually, I have to get it out of you before we move forward. You can guess this. So what is it that we need to build? What are the building blocks of protein? Because we talked about earlier, like the building blocks of starch is glucose, right? That's the monomer of glucose. The building blocks of nucleic acids are nucleotides. So what are the building blocks of these polypeptides? The proteins that we're about to build, what are they built with? And I'll give you a hint. And after this, hopefully someone really gets this right away. There's 20 of them. There's 20 different versions of them. Just like, remember for DNA, there's 4 different versions of nucleotides.
Enzymes.
Oh, so close. Not enzymes, but enzymes are made up of proteins. So what do we use to build polypeptides? And there's 20 different versions of them. And I'm just hoping and praying that some of you know this. You're just too shy to say it, because you should know this at this point. We've said it a lot.
Chromosomes.
Chromosomes are where we got the code from, but I like that you're at least talking about stuff that's relevant. Transfer RNA is bringing in the amino acids. That's what it's bringing in. That's what polypeptides are built from. That's like what we talked about Monday when I give you that big circle that says the code, how the code works. Like, you follow the first letter to the second letter to the third letter, and that says, bring me this amino acid. Bring me that amino acid, right? That's what we're after. We're trying to build a protein. We're trying to add amino acids, link them up one at a time. So that's what it's transferring. Anyway, this whole process requires an interpreter, and that interpreter's job is to convert the 3-letter nucleic acid words, as we call them codons, into amino acid words. So this part, this bullet point we already talked about, I think it was on Monday. So we already talked about that. Right? So we talked about how this language, the nucleic acid language, has 4 letters, right? A's, C's, T's, and G's for DNA, or A, U, C, and G for RNA, right? So the alphabet in that language consists of 4 letters. And then we said every word in that language is only 3 letters long, right? So it's some different version of how you put those letters together, and they have to be 3 letters long. So that part, that's a review. You already know that. Again, the new part here is this the transfer RNA, the tRNA, that's going to match the particular amino acid to their codon. So it is, in a sense, it's like, I don't want to say it's the translator, but that's the thing that's bringing in, like I said previously in Monday's lecture, I kept saying, bring in this amino acid, bring in that amino acid. So when I say bring in this amino acid, bring in that amino acid, this is the thing that's doing it. It's the tRNA. It has a code that matches its specific amino acid. And remember, there's 20 different amino acids. So, there's going to be at least 20 different RNAs. I could get more complicated, but I don't want to. Let's just say there's one specific tRNA for each amino acid. It has the code that it can help attach to that amino acid. And on the other end, it has the code that recognizes its word, so to speak, on the RNA. So it recognizes it on the RNA and said, oh, that's me. That's me. I'm the one who's just bringing the square amino acid. Or the other one's like, oh, that's me. I'm the one who's just bringing the circle amino acid. So TNA carries out 2 functions. It's basically what I just said, but let's read it from the screen. 1, it picks up the appropriate amino acid. So again, they're very specific. Each tRNA has its own specific amino acid. Out of the 20 of them, it has its own specific amino acid. And then again, it recognizes the appropriate codons in the mRNA. Again, using that example that we used on Monday where we said square amino acid, circle amino acid, triangle amino acid, which is not how it works, but it's a good visualization. That'd be like if I gave each one of you a different shape. Like I gave him a box, I gave her a ball, I gave him a triangle, I gave him a rectangle or whatever, and then I also assigned you a 3-letter word. And then I came up here and was like, all right, if you have CCG, I need your amino acid. And you would look and like, oh, that's me. And you would come and you would bring your amino acid. And I'm like, all right, if you have GGU, bring me your amino acid. So you would come and you would bring it. You, in that analogy, would be the tRNA. You have a very specific amino acid, the one that I've been given to you, and you have your one code. So once you're called up, you bring in your amino acid. Hopefully that makes sense. So, of all this, it is the unique structure of the tRNA that allows it to do both of those things. That allows it to attach to its specific amino acid, and that allows it to recognize its own code on the mRNA. And the next slide we're going to look at is going to kind of discuss that last, or visualize that last bullet point. But are there any questions about what's on this slide? Okay, again with the tRNA. Just so you know, I'm not going to ask you this, but tRNA is made up of a single RNA strand. It's worth mentioning, but I'm not going to test it now. But sort of like compare that to DNA, right? So we know DNA is actually, that double helix is actually made up of 2 strands of DNA, right? 2 strands of DNA that have been hydrogen bonded to themselves. But tRNA, despite having some hydrogen bonds, is actually made up of a single strand. So it kind of folded up upon itself. Again, I'm not going to ask you that. And because of this, like you can see in this picture here, it does form double-stranded regions, right? Right here, you can see they're hydrogen bonded to each other there, and here, and here, and here, right? So they do fold up on each other and form double bonds. But you don't need to know any of that either. The only reason I included this is because it was in your textbook, so I wanted to at least address it and tell you, hey, when you read that in the textbook, don't worry about studying it, don't memorize it, it's just so you know. That's how it gets its 3-dimensional shape, so it can do its job, right? And that's a big theme that we talked about in biology, right? The whole shape versus what it does. That's why your textbook includes that. So at the end, at the bottom here, you can see this is called the anticodon loop. And I'll also put it next to that. I'm going to describe what an anticodon loop is, but I'm not going to use that term in the exam. But let's talk about it. So at the end, like I just showed you, that's called the anticodon, and it is complementary to the codon on the mRNA. Meaning, if the mRNA is, I don't know, CCC, if that was the word on the mRNA, then the anticodon would be GGG, right? Because C's go with G's. So it would read that and be like, okay, if the RNA has, and this is just an example, but if the RNA has the codon CCC, then say the mRNA, then the tRNA would have the anticodon GGG, and it would recognize it, like, oh, that's me. That's where I need to come in. So that's at the one end of the RNA that I just showed you. On the other end, and I'm going to give you another better picture on the next slide, on the other end is where the actual amino acid attaches, which should make sense, right? One end, the amino acid attaches. The other end is where it reads its code. Here's a picture of that, a better picture of it. And again, I think this is not from your textbook. So this is a more realistic version of what it looks like, but I don't like it. This is, I think, how you should visualize it. Just simplify it. Right? In this course, you can just simplify it. So it's a thing, a tRNA is a thing that at one end holds an amino acid, and at the other end, it has an anticodon. It has a thing that just recognizes its code. It's like, oh, that's me. It's calling for my specific amino acid. That's all that is. And that's what a tRNA looks like. We can get through this. Ribosomes, that's the other part that we need. Remember, we're talking about the parts of translation. We haven't even started to talk about the process yet. So the other parts we need are the ribosomes. Ribosomes are technically organelles, which I'm not going to ask you about that. They coordinate the functioning of the mRNA and tRNA, which is another way of saying we have this mRNA, we have this tRNA. The ribosomes are simply the things that bring them together. And that being said, these are the things that actually make the polypeptides. Because again, remember, the mRNA says, hey, bring me this amino acid, bring me that amino acid. The tRNA says, oh, okay, that's me. I'm going to bring you the amino acid. The ribosomes just bring it all together, right? Here's the mRNA, here's the tRNA, here's that amino acid. I've got it. Bring me the next amino acid. I've got it. Bring me the next amino acid, right? What I might ask you, but probably not, is that ribosomes are technically made up of 2 subunits. Each one is made up of protein and one more type of RNA called rRNA. And that one's easy because it stands for ribosome. So the stuff that makes up ribosomes is ribosomal RNA and some proteins. But again, I'm probably not going to get that deep into the exam. And then now, at this point now, I've taught you all 3 types of RNA. It's a good time to end it for Wednesday. And the last word for attendance will be large. So I'll either see you in lab today if you want to be here, or online, or I'll see you on Friday. Coming to lab is optional, yes. Doing it is obviously not optional. Make sure you have it done by Friday. And it is really easy, but again, if you need my help, I'll be here and I'll be online.